It's just about time. So good morning, everyone, or good afternoon, depending on where you are. Thank you for joining us for this fourth webinar offered by the group Polyphenols. The group Polyphenols Society is an international association founded in France in 1972 with the aim of promoting research on plant polyphenols from their most basic and fundamental physico-chemical properties to their most diverse applications in food, agricultural, pharmaceutical, and cosmetic science and technologies. The group Polyphenol started to develop this series of webinars in Polyphenol Research in 2022, with presentations about polyphenols in soil system, food and beverages, and about their characterization. If you didn't have the chance to participate in the first webinars, you can still have access to the recordings on the website of the group Polyphenols. I'm Dr. Aude Watrelot, an assistant professor and extension specialist of enology at Iowa State University in the US, focusing on polyphenols, chemical properties in red wines. I'm the vice president of the group Polyphenols, and I'm co-hosting the webinar today with Professor Peter Constabel. Before I give the floor to Dr. Constabel, I would like to provide you some instruction. The webinar is being recorded and will be available on the YouTube channel of the group Polyphenol by the end of the week. Please type your questions in the chat box that you can find at the bottom of your screen by a click on the three dots. Your questions will be answered at the end of the webinar. At the end of the webinar as well, you will receive an email to complete a Quadric survey about how we did today and what we would need to change for future webinars. Please take a few minutes to complete it. Also, if you need a certificate of completion or participation to this webinar, please send me an email to request it. You will then receive it within a month. So, Dr. Constabel, the floor is yours to introduce yourself and our speakers today.
Great. Thank you, Aude. As mentioned, my name is Peter Constabel, and I'm really pleased to host this webinar. I'm at the University of Victoria, which is on the far west coast in Canada, and I'm a professor there in the Department of Biology. I teach there plant biochemistry and plant physiology, and my lab research is really focused on biosynthesis, regulation, and function of plant phenolics. So it's a real pleasure to now introduce our first speaker, Dr. David Ma. He's currently a postdoc at the University of Victoria, where he studies biosynthesis of bioactive benzenoid and salicinoid compounds. He has a BSc from Fudan University in Shanghai in China, one of the big universities in China. He did graduate work in plant molecular biology and biochemistry in my lab. And his PhD focused on understanding how flavonoid biosynthesis is regulated. And he's published some very nice papers in this area. He's worked a lot on the transcriptional regulation of proanthocyanidins and anthocyanins. And this is what he's going to talk to us about today. So Dr. Ma, the floor is yours. Please go ahead.
Can you see my screen? Okay. Thanks for the introduction, and thanks all for the invitation. So I'm going to talk about transcriptional regulation of anthocyanin and B-ring hydroxylation in poplar. So in this presentation, I'm going to first briefly introduce the anthocyanin biosynthesis and transcriptional regulation mechanism. And then I'm going to talk about specifically the anthocyanin MYB or MIB transcriptional regulators in poplar. And then I'm going to talk about flavonoid 3', 5' hydroxylase genes that are directly regulated by the MYB transcription factors. And at the end, I'm going to give a summary and also talk about some related research in the literature about anthocyanin biosynthesis in grapes, because I know the group, probably many of you study grape flavonoids or enology. Okay, so first part about anthocyanin biosynthesis and transcriptional regulation. So anthocyanin play a lot of important roles or have a lot of important functions in plant. So for example, they can, in the flower petal or the berry, they can attract pollinators and seed dispersal animals. And in leaves, they can act as sunscreens, especially in young leaves during the early stage of development or during the senescence stage in the autumn. And they can also act as antioxidant. So the anthocyanin are the end product of the flavonoid pathway. So the pathway starts with the condensation of p-crumoyl-CoA, which is synthesized from the general phenylpropanoid pathway, and straight malonyl-CoA by chalcone synthesis to make the A ring and B ring of the flavonoid. And then the chalcone isomerase will close the C ring. And then after that, it will add the hydroxyl group and then several reductase steps. I'm not going into the detail of the biosynthesis enzyme genes, but the most important genes I'm going to talk about are the hydroxylase genes. So the B ring hydroxylation, on the 3' position and 5' position will be catalyzed by flavonoid 3' hydroxylase or 3' and 5' hydroxylase at multiple steps at the early stage of the pathway. And they can take different intermediates like flavanone or dihydroflavanone as substrates. And the end product of the pathway include important flavonoids like anthocyanins I'm going to talk about. And Professor Scheer is going to talk about the proanthocyanidin or tannin biosynthesis. So the increased B ring hydroxylation of anthocyanin can shift the color of anthocyanin from pink color or light red color to red or dark red color, like cyanidin. And further hydroxylation by 3' and 5' position can shift the color to blue or even purple color. And the pathway is usually regulated by MYB transcription factor or MYB transcription factor. So the flavonoid pathway usually regulated by R2, R3 MYB transcription factor. So this is the phylogeny of flavonoids R2, R3 MYB transcription factors based on the protein sequence. And each panel here represents one subclade of R2, R3 MYB transcription factor regulate different end products. So including a subgroup of anthocyanin activators. So the R2, R3 MYB transcription factor, usually they have an N-terminal DNA binding domain with 2 MYB repeats, R2 and R3, and C-terminal activation motif. And flavonoid repressors, we call them flavonoid repressors because they usually have broad targets. So except for anthocyanin, they usually can also regulate tannins or flavonols. And they have a repression motif. And in the phylogeny, there are also R2, R3 MYB tannin activators, lignin repressors, and flavanol activators. And one characteristic feature of anthocyanin activators and repressors is that they usually interact with BHRH cofactor, and there's a BHRH interaction domain in the R3 repeat. So when there's developmental or stress signal, they can promote the MIP transcription factor or BHRH cofactor expression, and they form a complex and directly bind to the promoter region of the flavonoid enzyme gene and start the transcription of downstream flavonoid enzymes. And when the developmental or stress signal is removed, the repressors can then compete with the activator and bind to the BHRH cofactor and also bind to the promoter region, and the repression motif can repress the flavonoid enzyme gene expression and downregulated the production of the flavonoids. So specifically, the MIP transcription factors we found in poplar. So in our lab, we use poplar as a model species for flavonoid research. So first of all, the poplar genome, the Western balsam poplar is sequenced and is the first tree species with 4 genomes sequenced. And tissue culture and transformation methods are developed in several poplar species or hybrids. So this is a picture of tissue culture poplar in a magenta box. And the species is Populus tremuloides and tremula. So this is the main species we used in our lab for flavonoid biosynthesis research. And also in this species, in this hybrid, so the poplar accumulate high concentration of flavonoids in vegetative tissues, including leaf, stem, or roots. So this makes poplar a suitable model for flavonoid research. So we identified the poplar anthocyanin activators and repressors based on both sequence similarity and transcriptomic analysis of stress-induced poplars, including anthocyanin activators like MIP117. There are 5 paralogs of this gene in the poplar genome, but we choose MIP117 based on its expression is relatively higher than the other paralogs. And also flavonoid repressors, MIP165. There are also other paralogs. We also characterize those paralogs, but in this talk, I'm just going to focus on one of them. So to further characterize these transcriptional factors, we generate MIP overexpressing hybrid poplars. So we make this construct with double 35S promoter with the MIP transcription factor. So the MIP transcription factor can be constitutively expressed in the transgenic plants. So the phenotype of the activator, as you can see, when we overexpress MIP117, there's high accumulation of anthocyanin in the leaves, as well as the stem and roots. So basically all the tissue, all the vegetative tissue accumulate more anthocyanin. And if you look closely, the transgenics with high anthocyanin accumulation also show early senescence. And in comparison for flavonoid repressor overexpressing transgenics, you see, need to mention in the greenhouse, there's not much anthocyanin accumulation, so we put the transgenic plants together with the wild type control outside the greenhouse and exposed to the sunlight. And after 1 week of sunlight exposure, you can see there's anthocyanin accumulation in the young leaves, and this is more similar to the natural condition. Anthocyanin usually only accumulate in the young leaves. And compared to the wild type control, there's much less anthocyanin accumulation in the repressor overexpressors. And if you look at more closely of the color of activator overexpressing plants and repressor overexpressing plants, you can see the activator overexpressing plants, the leaf color is not only accumulate anthocyanin, but looks darker compared to sunlight exposed wild type. Then we did the transcriptomic analysis using RNA-seq to analyze the activator and repressor overexpressing poplars. And here it shows the fold change compared to the wild type of the activator overexpressing and repressor overexpressing poplars. So in the transcriptomic data, shows the differential expression of anthocyanin biosynthesis genes such as chalcone synthase, chalcone isomerase, and dihydroflavanol reductase, and anthocyanidin synthase. So these are all anthocyanin biosynthesis genes. And notably, we found poplar flavonoid 3', 5' hydroxylase is actually among the top differentially expressed genes. So in MIP117 or the activator of expressing plants, it's upregulated by over 150-fold compared to other biosynthesis genes. And in the repressor overexpressing plants, it's downregulated by 50-fold, over 50-fold. Just a reminder, so the flavonoid 3', 5' hydroxylase gene, it can add hydroxyl group on the 3' and 5' position of the B ring. So we speculate this may cause a change of the anthocyanin composition in the MIP117 overexpressing plants. So we did HPLC analysis to further analyze the anthocyanin composition in MIP117 overexpressing poplars and compare to sunlight exposed wild type. So you can see in the wild type, there's only one major peak, which is the cyanidin glycoside. But in the MIP117 overexpressing transgenics, you can see there are two other peaks. So we identified these peaks using SPEC, and they're actually delphinidin glycosides, and there are different glycosides with different sugar added on the 3 position. So the MIP117 overexpressing transgenics showed 54 to 340-fold higher anthocyanin content compared to the greenhouse wild type. And in addition to that, the delphinidin versus cyanidin ratio also increased over 10-fold. So it's not only increased the overall anthocyanin concentration, but also shift the composition or the profile of anthocyanin in these transgenics. So we think the flavonoid 3', 5' hydroxylase gene may be an interesting to study. So we further study the 3', 5' hydroxylase, and by overexpressing this gene in our hybrid poplar, and to see if it directly changed the anthocyanin composition. So we made a similar construct with double 35S promoter and the flavonoid 3', 5' hydroxylase gene. So the gene will be constitutively expressed in the hybrid poplar. So after we generate these transgenics, you can see the expression level of the 3', 5' hydroxylase is similar to the transgenic plants. It's about 50-fold to 130-fold upregulated in the transgenic plants. And we analyzed anthocyanin contents in these transgenic plants after exposed to sunlight. So there's in the transgenic, in the 3', 5' hydroxylase overexpressing transgenics, it also shows increased delphinidin. So these two peaks. And we measure the delphinidin and cyanidin ratio. So compared to the wild type, there's about 3 to 4-fold change in these 3' 5' hydroxylase overexpressing poplars. However, if we compare the 3' 5' hydroxylase overexpressing plants to the activator or the MIP117 overexpressing plants, the fold change of delphinidine and cyanidine is actually still lower. So we go back to our transcriptomic data, and we actually found the cytochrome B5 gene is upregulated by about 50-fold in MIP117 overexpressing poplars. And cytochrome B5 gene actually acts as a electron donor or the electron shuttle protein for cytochrome P450 or the flavonoid 3' 5' hydroxylase. So it provides one electron for the hydroxylase. So we think this might be the reason and related to the phenotypes that in the flavonoid 3' 5' hydroxylase overexpressing plants, the fold change is actually lower because there's not enough electron provided to the hydroxylase. So in summary, so we've found the transcriptional regulation networks of MIP activators and repressors in poplar, and overexpression of the MIP regulators in poplar affects not only the anthocyanin accumulation, but also change the anthocyanin composition by flavonoid 3' 5' hydroxylase. And after we overexpress the 3' 5' hydroxylase, we found so the hydroxylase is responsible for the B ring hydroxylation, but other genes like cytochrome B5 may also be involved and need further study. Okay, so at the end, I'm going to talk about some similar studies of anthocyanin biosynthesis in grape regulated by MIP transcription factor and 3' 5' hydroxylase from the literature. So grape actually is also a very interesting system because grape genome shows expansion of the anthocyanin MIP genes and also the 3' 5' hydroxylase. So in some cultivar like Cabernet Sauvignon, there are 16 copies of 3' 5' hydroxylase, and only one copy shows expression in the vegetative, so like leaf tissue, and the other 15 actually expressed in the grapes. And they're on one chromosome, suggests they may be duplicated very recently. And there are 3 MIP activators have been characterized and functional in grape. So the anthocyanin MIPs are called MIP-A in the grape, and they have different activation of the flavonoid 3' 5' hydroxylation. So only MIP-A1 can activate flavonoid 3' 5' hydroxylase. And because of the climate change, there's a lot of stress on the grape culture. So the UV light and drought stress actually can increase the anthocyanin accumulation in grape berries. However, the high temperature actually have the opposite effect on anthocyanin accumulation in grapes. So the high temperature can inhibit anthocyanin accumulation in berry skin via the MIP-A gene, and also the 3' 5' hydroxylase is one of the major genes that affect the reduced accumulation in the berry skin. Okay, so that's all for my talk. And acknowledgements, thanks to Eric and JP. We haven't talked about the hydroxylation change in the proanthocyanidin or tannin, but they also affect the tannin bearing hydroxylation, and Eric and JP helped us to analyze those tissues. And Michelle from Max Planck Institute helped us to confirm some of the structure of anthocyanin in the MIP117 overexpressing plants. And Peter's previous student, Hao, generated the flavonoid 3' 5' hydroxylase overexpressing transgenic poplars. Okay, and thank you for listening.
Thank you, David.
Yeah, thanks, David, for a really nice talk. And I would like to remind the audience that if there are questions, please type them into the chat, and we will answer those after our second presentation. So now I'd like to introduce our second speaker, Dr. Dayu Xie. He's a professor in the Department of Plant and Microbial Biology at North Carolina State University. He has a PhD in plant physiology from the Institute of Botany in the Chinese Academy of Sciences, and did postdocs at the National University of Singapore, as well as at the Noble Foundation in Oklahoma, where he worked with Richard Dixon, who's a really well-known plant biochemist and has done a lot of great work. Dayu's also who was a president of the Phytochemical Society of North America. He's a fellow of the Association for the Advancement of Science, and he's a co-editor-in-chief for Planta and Phytochemistry Reviews. He's going to talk about this today. One of his major contributions to plant biochemistry was a discovery of anthocyanin reductase gene and pathway, which is important for proanthocyanidin synthesis. And I still remember the paper when it came out because it really had a big impact on how we think about a proanthocyanin and how they're made. And he's also got some interests in metabolic engineering of flavonols and proanthocyanidins. So I look forward to your talk, Dr. Xie, and the floor is yours.
Peter, thank you so much for your last introduction, and also want to appreciate Dr. Watrelot for your invitation. And I'm so honored to give this webinar talk because I think this is my first time to give a talk in a group Polyphenols. I know Polyphenols group for a long time, but never got a chance to attend. Thank you so much for these opportunities. Okay, so today my talk is I call it the ANR pathway of flavonols and proanthocyanidins. So let me see. So I started with this one, acknowledgement. As Peter just mentioned that, I was a postdoc with Dr. Richard Dixon, and was so lucky, honored to be part of his lab and work on this project. And we got something published very nicely and was so lucky. So that's what today, part of my work is really my previous postdoc work. The reason is that I want to introduce something how discover ANR pathway. Something I want to share with you my experience, particularly for young students. And then I'm going to introduce some of the work I have been done in my lab, my own lab. And there's people here in my lab, and Dr. Sai Yozak, Dr. Yue Zhu, and also Min Zhu. I'm a 3 graduate students. They participated in some of the work I present today. So my talk is actually equal 2 parts, and I want to introduce you a little bit about before 2003, what was going on in the proanthocyanidin powered biosynthesis. There were so many debate when I was a student and also was a postdoc. Actually, we face a lot of challenges. So then second part is really since 2003. They particularly over the past 10 years, really 20 years, the progress in understanding proanthocyanidin biosynthesis is really dramatic. So yeah, here is just a summarize, and based on 2 years ago publication about the flavonoids, I want to share with you that where is proanthocyanidin in the whole polyphenol pathway and a particular flavor pathway. Hasling and Dr. Erber, their previous publication books I summarize here, and in the whole flavor pathway, we have summarized as 50 groups. And proanthocyanidin here, I summarize in number 8, okay? So here is, together with flavor 3,4 diols, anthocyanin, flavins, and proanthocyanidins. Last years, I want to add one more group. Last years, we published another group of proanthocyanidin flavonoids. We name as piperidins, as a group of oligomeric and polymeric flavonoids. So if you're interested, and you can look at this paper published in Molecular Plant in 2003. So give you some example proanthocyanidin. This is the monomers. It's major monomers here. It's flavor 3 ores. If I give you exactly examples, epi-aphrodisiac, epi-catechin, epi-gallocatechin, catechin, gallocatechin. And dimers here, like proanthocyanidin B1, B2, B3, B4, A1, and 2, there's 8 type of them. The reason I want to give this nice is that, and we are, when you look at literature, these 6 major monomers actually from the major components of proanthocyanidin. So far, we have identified more than 1,000 in the literature, okay? So as flavor 3 or proanthocyanidin, basically proanthocyanidin is really the oligomers, polymers of flavor 3 ore here. So it's really like this one, and consists of extension units, as many of you know that, and extract units. And also linked by very specific bands called interflavor bond between C4C8 or C4C6. I'm not really listed here. And another major features of proanthocyanidin is that after cleavage, used to be before 2003, I think most of book and textbook or maybe a reference book called hydrolysis. I found out that proanthocyanidin is not really soluble in the water. We have to really use organic solvent to extract it. It's not really soluble. So because proanthocyanidin is not water soluble and hydroxylation is soluble. So basically since then I changed my publication always as a cleavage. So if you use HCl butyl boronate, you cleave it into flavor bonds and also produce anthocyanidin. This is one of the major features, and we have known so many years. So that's the basic structure. And why we work on the proanthocyanidin, why proanthocyanidin will have been so a very attractive topic in the flavor pathway. One of the reasons is that they're as David just introduced that, and like anthocyanin, proanthocyanidin flavor 3 are very strong antioxidants, and many of you know that. I think it's one of the major components in many food, particularly in the nuts and cacao, those kind of grape, berry, those kind of agricultural products. Another one is really, there are so many research show that there is very good antigens for hemophilia leukemia. They have many antibacterial, antiviral functions, and also is they can rejuvenate people, hopefully have a rejuvenation, okay, for hair growth. And since I started my lab, I also start to work on the medicinal aspects of proanthocyanidin flavor 3 ores. And I want to share with you that over the past 4 years, we're lucky to publish a few papers about the flavor 3 ore like epigallocatechin gallate and epicatechin gallate and gallocatechin gallate. All those compounds have shown very promising anti-COVID activities, particularly main proteins. And we were tested at the coronavirus and alpha coronavirus 229E, which is called flu. If we found that, and it really flavor 3 or proanthocyanidin, dimeric proanthocyanidins, they're really promising in prevent coronavirus. So another major benefits, as many of you know, there is proanthocyanin is a very fundamental nutrition component in the animal forage crops. Because used to be when I was a postdoc, I was always told in Oklahoma because the cowboy that raise the cowboy over there every year they lose a lot of milk cow because the alfalfa cause blood diseases. And then USDA or in America and Australia or many other countries, New Zealand found that added proanthocyanidin can reduce the blood diseases, improve milk productions, and will increase weight gain, particularly reduce methane gas release. So that's why, and we all love, that's why and the proanthocyanidin is so important in both human health and also agriculture. So before 2003, this is summarize the Biogenesis pathway. One of the major, I'm not going to read every phase, and one of the majors at that time before 2003, that one of the major hypothesis is that glucosinate reductase was proposed as a key enzyme to catalyze glucosinate to plus catechin plus gallocatechin, which is called 2R3S23 transflavin not really epicatechin. And before 2003, and the plus catechin plus gallocatechin were claimed as the major dominant flavin compounds, particular stereochemical structures dominate in the literature. But we found that later on we found that it's not true. So, and the glucosinate reductase was tested many times in different publications that use glucosinate as substrate to produce a 2R3S23 transflavin. Most of the work have been done by radioactive labeled trace. Okay. Another major question is about how plant condensing flavin oil to condense tannins. Well, the major hypothesis that is plant use glucosinate extraction units, use flavin oil as a start units in the presence of condensates, which is proposed, and that form condensed tannins, okay, proanthocyanidin. That's a major thing. And I found that David just mentioned a very good point. Before 2003, they always considered that proanthocyanidin means that, proanthocyanidin in the before anthocyanin, okay? And that's the definition, used to be. And also claimed that anthocyanidin were claimed as the end of compounds of flavor pathway. That you just mentioned anthocyanin as end of compounds, one of compounds of end compounds of flavonoids. But anyway, so the major says, and that the progress before 2003 like this. So I'm not going to repeat more, but there's two fundamental work published in 1997 and 1999. As all of you know that in the 1990s, early 2000, and Rhabdopsis, even right now, is a major model plants to elucidate gene functions. And from France, Dr. Albert and David, they published two papers, 1997, 1999. The screen mutants of Rhabdopsis found the mutants called Banus. Banus is French. And they found out that as a wild type Rhabdopsis young seeds, you do not see any color, just greenish, yellowish, and because as proanthocyanidin, the flavor source basically is as colorless. But they found a mutant that if this mutant accumulates more anthocyanin, that means the enzyme mutant. So this is a phenotype of the seeds. This is they do the microscope observation. Say wild type, you do not see any anthocyanin, but the Banu has so many signs. So based on previous understanding hypothesis, look at anthocyanidin, they proposed that this band, they call band gene, and you call it protein, use glucosinate as a substrate to produce anthocyanidin. I produce flavin oils and then to proanthocyanidins. and compete with isocyanate synthesis. So that's the hypothesis. And after like 4 or 5 years function analysis, and nobody could test any leukoisocyanate reductase activities. So, and when I was postdoc, I was so lucky and work on this one. As we work on the medical trication at that time was a major project, function genomics work in Global Foundation. I was so lucky to work on a seed code EST stuff. So this is the red color now come from the proanthocyanidin reaction with vanillin HCl produced red color. Means that medical trication seeds is rich of proanthocyanidin. So based on previous hypothesis, we also proposed that leukoisocyanate reductase use leukoisocyanate as substrate to produce 2R3S323 chains, flavor-3 ores, and produce condensates. That's our hypothesis. Then we try to clone the gene and express your protein. Everything I was lucky, I got it. And we clone the gene. Leukoisocyanate, the gene Benegene. We got a Benegene, and we induce a protein. This is a protein over here. And then we feed the leukoisocyanate as substrate, radioactive labeling, everything we tested it. And we, in the presence of NADPH, NADP, NADH, and many other reductors, coenzyme. And what I want to share with you that particular for student here, I almost did more than 700 reactions, all type of reactions. I get use radioactive everything. I get a 50% positive, 50% negative, which is in the presence of my protein, leukoisocyanate look like it's converted to the flavor-3 ores. But 50%, I have more than 300 reactions give me positive. But at that time, I cannot cheat myself. I said, if I publish it like that way, nobody will question me because I have more than 300 reactions repeat. But how about another 50% active results? So I was thinking, I debated with some advisor, Richard Dixon, said, no, I'm not going to believe those things. And also skeptical about the activity. So then I stopped experiment. I read a lot of literature. I found that I know that time, all previous book claimed that isocyanate is what type of end products in the flavonoid pathway. That by that saying that, by that side, means no other opportunities to use isocyanate as substrate to make another compound. At that time, I found that then no matter what I try, Ben does not work in this way. The hypothesis is that, okay, maybe use the isocyanate reductase, use isocyanate substrate. It's very easy to test because I have an enzyme. I just feed it my enzyme with isocyanidin. And I have that time, isocyanate is very, the commercial available purchase and feed it just 20 minutes. Then we got a new compounds like here is really like a product 1 and 2 is a P1, P2. But all control, there was nothing over there. So then I was very also skeptical about this one because everybody previous me in the previous claim that isocyanate as an end product. So then I use panagonidin as a substrate. The reason is that time there was no commercial available FF-Zelakin and in the market. So if I can get a new compounds, people are not skeptical about me. So then I use substrate. I also got 2 compounds, and compounds 1 and 2. And then we use different masking, RC mass, GC mass, and more, and circular dichroism to profile and demonstrate that one of the major one is minus FF-Zelakin, which is 2R3R236, and on the trihydroxyflavor-3 ores. So we are very happy for that one. And then we found on that. And we were very cautious, particularly Dr. Richard Dixon was very cautious. And at that time, I named this enzyme as isocyanate reductase. So then we try to use this enzyme from Rhabdopsis, and I use different substrate. There, everything is correct. I mean, so we can make a 10 milligram, 20 milligram, even though, 100 milligram. So by that time, so there's nothing that's the data. So then we name this enzyme as isocyanate reductase. And finally, we can characterize all those. We published this one later on about rhabdopsis, isocyanate reductase, and metacargo reductase, and kinetics. I'm not going to read this one. So then we this is in vitro, as all you said, if in vitro, and that does not mean in vivo. So I overexpress in tobacco, because tobacco is so easy to do. So anything you can, this is different transgenic tobacco plant flowers. You can see the flowers, different lines, different transgenic events with different copy number of transgene. And found out that they all become white. And this is C5 is control, and also 1-to-1 is transgenic control. And also use dimethylaminosinamide stain that you can see the blue color. Blue color result from dimethylaminosinamide reacted with proanthocyanidin. And also we can use microscope identify this blue spot really look like say in the vacuum. So this is things, but this is not really enough. And we just want to show whether or not this is transgenic does not mean is really that cannot replace genetic data. So, and at that time we're trying to get genetic data. Luckily, we had a banner mutants, this wild type. We proposed this pathway from ANS to ANR and to flavicol proanthocyanidin. And then, okay, banner mutant here, we know if we lock it out here, lock out this one, we have a banner mutant that produce more isocyanate. And we need one mutants if we lock at the ANS, the whole state should become transparent. And that time we got a TT18 mutants, the states you say become transparent. So put all together, genetic data together, and prove that, and the pathway is here is proanthocyanidin new pathway from ANS, isocyanidase, ANR, and to flavicol or proanthocyanidin. So that's the thing. So one of the main, we were so happy for that, but one of the major, this project is for application, agriculture. So we want to see whether or not this pathway can be used as a protein to engineer proanthocyanidin for our animal nutrition. So as many of you, if you know Metacargo trichacula, Metacargo is a beautiful, beautiful and forage crop because they have a single leaf in the center. There's a red spot of isocyanate. So with then overexpression in R in the Metacargo trichacula, we got different transgenic lines. You can see this is a control. This is control plants. This is all my transgenic plants. Definitely, they produce a new and dominant oligomer proanthocyanidin in Metacargo trichacula. That is that when you apply this pathway in the plants, which can produce isocyanate as a substrate. So the second question is that how about the plants? How about the tissue that don't have isocyanate pathway? So and with that time, we're trying to find a new gene, and David just mentioned about the MAP 117 from the poplar trees. At that time, actually, our lab found the new genes called PAP1 is R2, R3. Thank you, David, for introduce about R2, R3 family. So PAP1 also R2, R3 family. Right now, PAP1 has been demonstrated as a master regulator. And I know there's so many argument, debate about PAP1 functions. Yeah, our previous work that is very crazy that PAP1 overexpression, activation, and rhabdopsis, they do activate most of gene expressions, increase that, okay, increase. And that's why we draw this one. I know many people found that major focus this gene and only a major upregulate or activate the pathway, later pathway gene like DFR, ANS, and the glucotransferase, but not ANR. But if I depend, my argument is that depend which material you select at the time is so important, okay? So we test different materials, different time, we found that PAP1 does increase almost every single gene expression if you find the right time. So then, okay, we propose to use PAP1 as a regulator to activate the anthocyanin pathway. What we did that, okay, we tested tobacco. Basically, tobacco leaf is evergreen, okay, it's or may become yellowish, but never produce anthocyanin. So when you overexpress PAP1 in tobacco, this is my genetic breeding, I got a super purple tobacco plants will grow the field. It's no gene sinus. It's about almost 20 years, no gene sinus at all. So actually in the right figure show you that this is anthocyanin, they only produce in the flowers, but not in leaf. But anyway, so and our all transgenic plants produce darphene, panagalin, and cyanin. Okay, show the different peak over there. So active the whole pathway. So what I did is that I have a transgenic tobacco, ANR, then I cross PAP1. So we get a PAP1 ANR transgenic tobacco class. So our hypothesis is that if two genes collaborate to work together, PAP1 ANR transgenic should lose anthocyanin. What we found out that this PAP1 ANR, they lose anthocyanin compared with PAP1 alone, but still produce some anthocyanin. So then we do the TRC analysis. You see this is here, this is transgenic PAP1 materials you can see produce different proanthocyanidins and also flavor-soluble oil over there. But the control, you can see PAP1 did not, PAP1 vector control transgenic, but they don't. So that means that this result shows that, okay, so is the ANR pathway definitely has some meanings, importance for metabolic engineering of flavor-soluble oil and proanthocyanin in plants. So, and by that time, and later on, that we face a lot of challenges there. So, because this is different from textbook and reference book previously, we all think about it. So many people challenge us whether the ANR's pathway evolution conserved in nice plants, in the plants, and is the ANR pathway co-evolved with the RAR pathway? So when I started my lab, I started trying to address these two questions. Of course, as genome sequences more and more and available in the market, in the database, easy for us to look at it, use phylogenetic tree to do that. Definitely, this is the simple phylogenetic tree. This tree just show you that, okay, most of plants genome sequence, we can find an ANR, okay, and also a flavor-soluble proanthocyanidin. So in addition to do that, and also we developed a method, very simple method to analyze proanthocyanin pathway in the plant kingdom. Just to do the ANR as biochemical enzyme, relatively easy to extract. We developed 5 different protocols to extract the ANR enzyme: TRC, method, HPLC, RT-PCR, phylogenetic tree. So what I will show you that we did all those things. We did a phylogenetic tree. We selected plants from all the way from fir plants and angiosperm, gymnosperm, all these plants over there. We developed 5 different protocols to isolate ANR to test activities. You just need simply testing it. You can use TRC or you can use HPLC to detect epicatechin, minus catechin, or those compounds. Definitely all those methods demonstrate that the pathway is preserved in the plant kingdom. So it's really That's the main thing. And for the past years, a lot of progress after I left Richard Dixon's lab, including my own lab, and also major progress in his lab. I want to share with that many new things. Possibly takes like possibly 2 days workshop to introduce you everything. One of the things that he found the new activities, LAR, leukocyte reductase, new activities. And the other thing is really my work. When I collaborate with Dr. Xia in China, we use approach to demonstrate the flavor carbocations. We know that carbocation we propose as an intermediate, which involved the polymerization of proanthocyanidin. But nobody really tracked it. If you look at the literatures, how can you track that? How can we see that? So we found a way to track these carbocations and intermediate. So we published a small paper. And later on, my Dr. Richard Dixon found that ANAS, there is the diverging deoxygenase, so which have very different functions, and they use catechol as substrate to produce anthocyanidin. It's it's really unexpected. And later on, he's the news, not Dr. Zhu, and follow my work and found that anthocyanin reductase has another functions, and in the proanthocyanin dual functions, and also they also continue found that as a conjugate of catechol plus catechol as extension units, and for proanthocyanin biosynthesis. So it's really because of time issue. I only can introduce this kind of fundamental work, but I'm going to summarize there. Right now, proanthocyanin biosynthesis is the pathway here. In addition to all this black arrow here, the ANAS and the ANR to here, epiflavin ores, and also and the leukocyte reductase produce flavin ore plus really plus catechin. And the red arrow here is a new discovery in the last 10 years, actually 8 years. Why is leukocyte reductase actually use flavin ore for sulfate cysteine acid as a substrate to produce plus flavin ores? And the leukocyte deoxygenase, which is a homologue anthocyanin synthesis, is really not just homologue, it's a paralog. But its function diverging, it use plus flavin ores as a substrate to make anthocyanidins, then go to anthocyanin reductase to produce and proanthocyanidin. So it's very, this is the evidence here is how anthocyanin reductase works. Anthocyanin reductase use this one and flavin 3 and 3,4 diol, and flavin 2 and 3-ol. These two compounds super unstable, so you have to check them and use RC mass fragmentation, identify them, and use substrate to produce and minus epicatechin minus epicatechin. So all this work here. So and that's really to me is very exciting and very new information. And all this information showed us that it's really the biosynthesis polymerization of proanthocyanidin is more complicated than we previously hypothesized. So now in the current revision of the flavin and the PE biosynthesis, and here or here, I was trying to summarize here, flavin for sulfate cysteine acid use substrate catalyzed by LAR to produce flavin ore. Flavin ore catalyzed by LDUX to leukocyte, then go to flavin ore that way. Okay, so the analysis is that we recently found that flavin ore actually is another substrate for oligomeric heparidins, which is very similar like a proanthocyanidin, but the skeleton is different. So actually that's my introduction. And in a summary, the take-home message for you, and the NR pathway is conserved, conserved in seedlings and seeded plants, include fern and gymnosperm and also angiosperm. And definitely metabolic engineering use is right now very successful to use anthocyanin pathway, anthocyanin reductive pathway to produce flavorful or processing in plants. And the news is that from my supervisor's lab and the ANR area, RDOX, they have a dual functions in the biosynthesis of flavorful oils. So it's really future work in these areas. In addition to that, I know there's argument debate about how plants condense proanthocyanidin. I still hypothesize that a condensate exists in nature. The reason is that every single plant produces different format, stereochemical structures of proanthocyanidin. If really the chemical synthesize, they're supposed to 50%, 50%, but it's not really, okay? Some plants majorly consist of, one type of stereochemical structures. Another plants make another stereochemical structures. So it's really still a lot of work to do trying to elucidate the mechanism by which how plants and the condensate proanthocyanidins. So, yeah, to the end, I'm stopped here. I really appreciate your attention. Thank you so much.
Great, great. Thanks so much, Dayu. That was fascinating, and I really appreciate the historical perspective on how this field has changed. We stumbled on a co-expression of the anthocyanin synthase way back, and it co-expressed with the proanthocyanins, the tannins, and we could never figure it out till your paper came along, and then it all made sense. So it was quite amazing. So. Yeah, a reminder to the audience, please put questions into the chat. Maybe we should start with the last one. I just see something about any prospect for commercialization of this kind of research that you, and specifically with regards to nutraceuticals of plant polyphenols. Maybe you could just go with that question.
Yeah, I gave us three. I was attacked by people when I reported 2020. We reported EGCG or other flavorful proanthocyanidin can inhibit MAY protease to prevent COVID. I was attacked by people. They said I was the person who was against vaccination. I was no, and it's crazy actually. Later on, I stopped. There's so many TV want to interview me. I had a full interview, and I found that the publicity was terrible. I was not good at it at all. So I stopped all those things. But I did a hypothesis that by that time, I gave a talk in the PSAA. My hypothesis is that if everybody drink high concentration or take a 300 milligram flavorful or proanthocyanidin per day in the world every day for 1 month, COVID is supposed to be stopped by mid-2021. That's my hypothesis. Later I published in other papers about green tea. I have more than 200 million people data. I found that I use green tea as an example, drink more green tea and lower death rate. Particularly Turkey. Turkey has, since the beginning, their death rate lower than 1%. Turkey has the highest tea consumption in the world. Every year, everybody, every day, they drink almost average 10 gram tea. So I calculate that they take possibly, I guess that possibly 500 milligram those compounds per day. The oldest lady is 96 years old. She survived from COVID by not taking any medicine, just the green tea. He survived 99 years old. He passed away not because of COVID, because hurricane. Because the hurricane is conditions. His grandson told us that. So my question, my commercialization definitely has a bright future. Many years, right now I'm working on the medical science of this site. I found it super difficult. The reason is that many big pharmaceutical companies, they do not want to do that because everybody can get it from nature. So the pharmaceutical company will not support because they cannot get a profit. So that's our research is very hard to get. You cannot. It's hard to get a grant, but a commercialization definitely is there is a bright future. But I can share with you that American Natural Products Society, natural products is not by scientists, by just the most public nutrition science. Their revenue $65 billion per year just to sell polyphenol or other compounds. Okay, over the past 10 years, almost average $15 to $65 billion per year. So even though those big pharmaceutical companies do not want to do this, we still have many folk medicine and many nutraceutical companies that work very well. But simply say that the commercialization aspect, I think, has a bright future.
Sounds good. Yeah, I know it's tricky. Yeah, thanks for that. Maybe we'll go to an easier question for David about how the fold change was calculated in the transcriptomic data. When we calculated the fold change for the hydroxylase.
Oh, okay. Yeah. So we do have standard for cyanidin and delphinidin glycosides. One thing I want to mention is that we only have standard for cyanidin glucoside and delphinidin glucoside. And the two peaks I show for delphinidin, they're actually different sugar add on the 3 hydroxyl group. So they're delphinidin glucoside and delphinidin semi-bioside. So it's a cellulose and a glucose add onto the sugar, but we only use the cyanidin glucoside as the standard. And the UV absorbance should be mainly on the cyanidin. So they should have very similar standard curve. So we just generate a standard curve with our standards and calculate the concentration and calculate the fold change.
So can I clarify just quickly that question? Would you be able to explain a little bit more how you are able to calculate how many times that specific gene is upregulated? How do you calculate that?
Oh, the gene expression, sorry. Mm-hmm. The gene expression, so we use, I guess you mean the fold change in the RNA-seq data. So in RNA-seq data, so basically it measures how many transcripts or fragments or transcripts in it expressed in the transgenic plants. So the fold change is basically how many transcripts of the specific genes in the MIB overexpressor or repressor transgenic plants, and divided by how many transcripts in the wild type.
Yeah, there's a follow-up there. Maybe we should go to that. The relationship of gene expression to end product quantity. I guess that was some of your data that you brought up, David. Do you see that last question?
Yeah,
Relationship of that data with the end product.
Because the end product is actually affected by many genes, right? It's not only affected by one gene. So I wouldn't say it's linear, but the overall trend is the same. Like, so in the activator, when many of the pathway genes are upregulated, the overall products are also upregulated.
Thank you.
Yeah, I was going to ask you a question about that condensing enzyme. I guess it's very hypothetical. Is there anything further that you can say about what you're speculating, what it might be, and where that's going to go?
That's a great question. Actually, previously when I published a review paper, we proposed three group of enzyme, peroxidase and lactase, and also polyphenol oxidase. I think I still believe, I still hypothesize three group of them are really the major candidate. And that's basically what we found that is flavorous or polymerase. We originally I hypothesized that enzyme is supposed to be like a condensases, but we got in compounds, we purify compound, we found that after in the beginning we claim it's a proanthocyanidin, but after we figure out, like spend almost 8 years use NMR figure structure, we found that it's not. So it's basically the data we publishing about flavor polymerase. This is a type 3 peroxidase. I still hypothesis that maybe one of the peroxidase and lactase polymerase and polyphenol oxidase involved this one. The reason is that, one of the other reasons is that I can share with you. If you look at all previous literatures, I said all, possibly I read 90%. So when you look at the data, very detailed, people publication details, there's so many information lack over there. People all claim the different polyphenol oxidase, but the previous, like before 2020, before 2000, most of people not get the details about the structures and also nucleotide or the amino acid. So there's so many detailed information that we do not understand. So that's the reason that we have not really found a candidate. Like a peroxidase, we have type 1, type 2, type 3. But this time we found it's type 3. It's really not many publications about type 3 characterization. So actually it's a big one after more genome sequence coming. So polyphenol oxidase also we just call polyphenol oxidase. We know that, we also claim that, or there is a localized in the chloroplast, but we found that they're actually localized in the vacuum. So that we need to look at those candidates, find a candidate, and whether or not they're really the same as we previously proposed that. Actually, one of my friends, he claimed that he found one. He found one of the lactase. So it's really, he asked me advice, suggestions. I look at it from his genetic data, so strong. He has a very strong genetic data. Look at one gene lactase and the proanthocyanidin geometrically reduced. So it's really, but he has not really submitted his manuscript. I also I can share with you. So I still hypothesis that this three family, all family can do this, but you need to find a way because one of the major thing is the carbohydrate. The carbohydrate format, and the flavin 3, flavin 2 and 3-ol, flavin 3 and 3-ol. That two compounds play very fundamental activities. So these two compounds decide which way will go. And very unfortunately, these two compounds is very hard to purify. That we got a little bit, we purify a little bit. It's there. So yeah, I'm still looking for that. Let's work together.
Yeah, I had some ideas about that too, but we can probably take that offline. There was a question there about the class of anthocyanin. This is for the first speaker, for David, about which particular class of anthocyanin and LCMS you focused on, and also if you know which mode for ionization was used. I don't know if do you understand that question, David?
Yeah. What class of anthocyanin? I'm pretty sure, but we mainly measure cyanidin glucoside and delphinidin glucoside. And I think we used a negative mode for the LCMS.
I think the anthocyanin profile in poplar is very simple. Like there's mostly cyanidin and some delphinidin, I believe.
Yes, I can add a little bit here. We work a lot here. I want to share with you the who asked questions. Look at all isocyanate or isocyanidin. We provide so many. What I can share with you that no matter which negative or positive, they always lose 2 proton. For example, isocyanidin molecule is 287. So when no matter which way you do, they lose. If you're active, you're going to have 285. No matter what you do, okay. But positive, you only can like a 286 because they always lose because there's a cation actually. It's a flavin is a positive charge is cation, so that part is easy to be removed. So that's what you only can get a major fragment has 2 proton lower than the molecular weight.
Right. Yeah. Thanks, Dayu. Yeah. There was a question about microalgae. Are these pathways specific for plants, and can they be transposed to microalgae? Yeah, that's a difficult one. I don't know.
Actually, there are flavonoids, I think, from the literature. There are flavonoids from microalgae, but they're quite different from the higher plants. And the pathway, some of the enzyme might be similar, but usually in the lower plants or like microalgae, the enzymes, they usually have very broad activity compared to higher plants. They have specific activity on specific substrates. But I've seen also like people use higher plant enzyme put into like yeast to produce flavonoid. So I think the problem is if you have like a transformation system or expression system for your specific species of microalgae to put this specific to test those specific enzymes in the microalgae.
I mean, the other aspect is making sure that you have enough of the precursors. If you can, in theory, you can put the genes in, but you need the primary precursors to feed into that pathway. And so that's often a limitation.
So David, I have a question for you. 2 questions. One, did your F3' 5' hydroxylase and flavor hydroxylase use camphor as a quercetin as substrate?
Yes, actually we did measure. Actually, JP helped to measure the flavanol composition, and we did see increased. So the most abundant flavanol in poplar is quercetin, and quercetin glucoside, and we did measure myristicin, and there's also like 5-fold increase of myristotin in the transgenics. And I want to mention the other thing is, so compared to anthocyanin, we also measure tannin, like per cyanidin and per delphinidin ratio. So compared to anthocyanin, which shows 10-fold increase, flavanol and tannin showed relatively lower increase of fold change.
Mm.
My second question is about the flavor 3,5 prime hydroxylase, and you use CYB5, because what is Q450, and both of them form actually from a complex partnership as electron transfer. So what did you realize any ratio, like a protein levels or maybe gene levels? What the ratio? Because there is a long-term questions that CYP need some CPR as a partner and to perform those reductors. Did you see any like ratio related data? For example, Q450 need a 5 or CYP just 2 or 3, something like that.
Actually, I didn't mention in my talk, but we did try to overexpress the cytochrome B5 gene as well, together with the flavonoid 3' 5' hydroxylase. And the cytochrome B5 gene, it actually has a much higher basal level in the plant compared to the flavonoid 3' 5' hydroxylase, the P450 gene. And when we try to overexpress this gene, we can't reach very high fold change compared to the wild type, but when we try to overexpress the cytochrome B5 gene, it's only about like 3 to 5-fold increase in the transgenic. And eventually we didn't get more delphinidin compared to only the hydroxylase overexpressing.
Mm. There's a question about downregulation, and that's for the first speaker, for David. If you maybe you can answer this one. If you downregulate a repressor, does it what happens? I don't know if you have data on that. I think we tried that. Theoretically, downregulating a repressor should increase the final product, right?
Increased, yeah. So we actually did, so that's by a previous Peter student. We knock out or downregulate the repressor, which may increase the end product.
Yeah, I think RNAi of some of the repressors, but I'm not sure they showed a phenotype, right?
Yeah, I did try to do the RNAi. At that time, there's no CRISPR method, but yeah, we didn't show we didn't see very strong phenotype if we downregulate the repressor by RNAi. I think the one problem is, like I showed in my talk, there are many paralogs of the repressors. So if you downregulate one of them, it won't show a strong phenotype of the end product, because there are other paralogs may compensate the downregulation.
Mm. Yeah, it seems to be all, in some cases it's more of a network too, right? And so there's lots of redundancy and maybe some compensatory sort of reactions. So not so simple. There's a question about polymerization and the acidity of the vacuole. I guess that's for you, you.
Yeah, that's a great question. I think is first of all, Asa, you're right, and in the acidic conditions, definitely the polymerization is better, and that exists in nature and also experiment. In our paper published in Plant Journal, actually, we already we report about polymerization without enzyme, because in the acidic conditions, actually, is a flavor carbocation is easy to form. That's how we use fluoroglosyrol and hydrolysis analysis of proanthocyanidin, because in the acidic conditions, particularly methanol, HCl, we can relatively easy to detect, and epicatechoic cation and catechol cation. So I think it's in that conditions, actually in acidic conditions, actually carbocation is not that unstable. We did purify a little bit by TRC, use acidic conditions. It have to be it's acidic conditions. If you move to the basic conditions, they're gone, okay? But it have to that we maintain that conditions, and we can see them, and we can use RC mass to detect them, and also we can use TRC, which is so insensitive method. We can scratch the silicas and can purify a little bit, and but we couldn't really do any NMR, because that definitely takes time, and then during the process they're gone. So yeah.
So when you say acidic conditions, that's like vacuolar pH 5 or something, or somewhere in that range?
No, it's if you, I don't know, you have experience. I had it for grape. David just mentioned the grape. Actually, grape skin, the pH value is lower than pH 1 or 2. The reason I can share with you that I measured it after you extract it, not use buffer, just use water to extract that. It's really the acidic conditions, pH value is very dependent where you grow your grape, okay? When I got a grape from Oklahoma, so it's really, the reason is that I can share with that when I isolated isocyanate reductase from grape berries, no matter what I did, I couldn't get any proteins. So finally, I found that the extraction, I use pH 7 trace buffer. Is no way to maintain a pH value to pH 7. I had to immediately add sodium hydroxygen by pellets over there, then adjust the pH value to pH 7 to get isocyanate reductase. So it's a pH value actually in the grape skin is very, very low. So I don't have exact data, but at least my impression is lower than 2.
Wow. Okay. Yeah.
Yeah.
Huh. Very, very interesting. Yeah.
Thank you very much, everyone. We still have just 2 minutes, so let's finish here. If you have further questions, please send us an email and we're going to share with the speakers. So just to finish, I would like to thank our fantastic speakers today, Dr. Ma, Dr. Xie, and our moderator, Dr. Constabel. Also, thanks everyone very much for your participation, and we hope that this webinar was helpful and brought some new knowledge to you. Please, as I mentioned, send us an email if you have any further questions for all our speakers today. So the presentation will be available free of charge on the Group Polyphenols website, on the YouTube channel of the Group Polyphenol, the Instagram and Twitter account of the Group Polyphenols. So there are a lot of different options for you to have access to that recording. Before just closing this webinar, I would like to remind you that you will receive an email with a survey. So please take 2 to 5 minutes to complete the survey. Also, stay tuned for further webinars that are most likely going to occur in July 2024. Also, other announcements. So there is the International Workshop on anthocyanins and beta-lines that is hosted at the University of Leeds in UK from September 17 to September 19th this year. So feel free to register using either the QR code that you have on your screen. Otherwise, you can have access to that event or that workshop on the website, the conference website. Another announcement is just for you to mark on your calendar that the next international conference on polyphenols is going to happen in Turku, Finland in 2025, in July 2025. So please mark your calendar for that. Thank you very much once again for attending, and we hope to see you soon. Thank you, everyone.